Thank you all. You're in for a treat. All of us sitting in this room are eager to hear about what's new, what's changed, and what, most importantly, is the impact going forward for anyone who's been touched with breast cancer. As advocates, we are looking for progress, not just change for change's sake. Please keep in mind, as you listen today about the many hypes from the past, there's never a silver bullet. In reality, the more we learn, the more complicated the science becomes. For example, from the promise of gene sequencing as the ultimate answer to the existence of epigenetics, proteomics, metabolomics, everything you can't pronounce, all additional layers to understand and unravel. This should form a background for all of you today to question the newest directions of research. As you've heard, Artemis is based on the concept of directing researchers' focus towards two areas of primary concern: prevention and metastasis. Has it made a difference? The focus on immunology did not exist before in breast cancer research. The DoD program, and specifically the advocates on that program, had a lot to do with recognizing and pushing for and supporting funding for immunology research. And that program has really led the way in making immunology a focus. There has also been more focus on metastasis, more than before, although it's hard to sometimes quantify exactly how much is directed and what's actually happening. And we also need to lead the way in ensuring that that work goes in the right direction. What is the importance of the Artemis for advocates? It is a model for advocate involvement in research. I would like to describe briefly the projects that our advocates have undertaken to inform the progress of the Artemis project. A group of advocates took on researching breast cancer prevention trials to date, both vaccine and non-vaccine trials, as well as in other diseases, such as diabetes and HIV. What can we learn from these prior examples, and how can it inform our path? Another important advocate initiative was the Biospecimen Study. Advocates set out to determine biospecimen needs for Artemis and interviewed 8 researchers to figure out what are the existing resources, what kind of tissue is needed, and how can we facilitate collaborations. Finally, a third group of committed advocates dug deeply into a published list by Dr. Mac Cheever of 75 known cancer antigens and did an extensive literature search to come up with 6 of the most promising targets for a breast cancer vaccine, present in the majority of breast tumors and associated with early breast tumorigenesis. The list was essential in the subsequent discussions on the development of a preventive breast cancer vaccine. We should all be proud of the accomplishments of our committed advocates who have, as I have shown, are collaborating as partners in new and significant ways. The question remains, however, there are how many of these, not necessarily NBC advocates, who else is out there challenging and collaborating with scientists and researchers in order to change the landscape? Today, we're going to hear from the best and the brightest researchers about their specific areas of expertise in designing a preventive breast cancer vaccine and affecting overall survival through the control of metastasis. All 3 of these researchers are members of the Artemis projects on vaccine development and metastasis research. We'll listen to a presentation from each of the panelists before our question and answer session. Please write your questions as they occur to you on the cards on your tables and hand them to the staff circulating in the room. We'll try to get in as many as possible, and it's real important. First, Dr. Keith Connexin is director of the Cancer Vaccines and Immune Therapies program at the Vaccine and Gene Therapy Institute of Florida. Did you come up with that title? It's a long one. Dr. Connexin was the director of the Ovarian Cancer Immunology and Immunotherapy program at the Mayo Clinic, Rochester, Minnesota. He focuses on the immunologic issues in preventive vaccine and therapeutic applications. Immunology is the most complex set of interactions, demanding a new vocabulary, a new set of assumptions, definitions of efficacy, trying to find markers of immune response and endpoints. At the same time, many researchers, most recently at the AACR conference in San Diego just a few weeks ago, have come to believe that immunotherapy holds the greatest promise for treatment and an eventual cure for breast cancer. And we at NBCC can take credit for that change in the conversation. Dr. Pat Stig is Senior Investigator and Deputy Chief Women's Malignancies Branch in the Center for Cancer Research at NCI. She has focused her studies on breast cancer metastasis suppressor genes, and site-specific metastasis to the brain. Many of us know Pat as one of the best teachers at Project Lead. She can explain just about anything, even PARP inhibitors. She will set the stage first with a sobering evaluation of the hard truths that metastatic patients face today, and then will describe some new directions in clinical trials for these patients, emphasizing new endpoints to define efficacy in the metastatic setting, and thinking always out of the box. Dr. Alana Whelm is an associate professor in the Department of Oncological Sciences at the University of Utah, an investigator at the Huntsman Cancer Institute, and a member of the Cell Response and Regulation Program. Dr. Whelm's group has developed new models to understand breast tumor growth and metastasis. Alana will talk about tumor dormancy, which is a primary research question for the Artemis Metastasis Project. Here is an example where prior scientific research is very limited, and Artemis has taken on the challenge to define tumor dormancy and its role in preventing metastasis. We advocates have to step up and challenge not only the gaps in scientific research, but also the direction of future work. We need to remember the goal of eradicating breast cancer. And during this session, I would like to challenge somebody. I would like to challenge the speakers, because there's a lot to think about, and listen, to one, explain why their work will not be one more decades-long path with only incremental progress, and two, what changes in the conversation, for example, in understanding the immune system, endpoints, do advocates need to prepare? And I will challenge the advocates as well in the audience to ask challenging questions to ensure that the researchers feel the same sense of urgency we do. How can this work be accelerated? What else do I need to learn more about so I can explain it to others? Are we all on the same track working towards deadline 2020? Advocates have their work cut out for us to encourage new ways to pursue that goal and to help translate new directions in research to the community.